Hi, my name is Paul Fisher from University of Florida, IFAS Extension. What we're going to talk about right now is 4 different types of testing methods to test root substrate in containers. We're going to cover saturated medium extract or saturated paste extract, the plug press or plug squeeze method, the 1 part of substrate to 2 parts of water testing method, and the pour-through method. All of these are very simple and practical methods. And I'm going to start with the saturated paste extract method, which is the method that's used by most of the horticultural laboratories in the United States. And they use that method because it's very standardized and has a very consistent amount of water mixed in with substrate. So what we do with the saturated paste extract method is we're going to collect samples from several containers. So typically from 5 to 10 containers. And so what we're going to do is we're going to take a soil sample out of the bottom 2/3 of the container. And as we explained in the lesson, the reason we're going to be in the bottom 2/3 is that's where most of the root growth typically is. And because of evaporation from the soil surface, we can get a higher salt level close to the surface. So if we took a sample from this area around the surface of the substrate, we'd probably get a high EC and a low pH, which would not be representative of the conditions that the roots are growing in. So we're going to take samples from several different containers. We're going to combine them into one sample. If we were sending that sample off to a lab, then typically they would want to have 2 cups of sample, or about 500 milliliters. So they've got enough sample there to measure all of the essential nutrients. So what we do is we combine our sample, and then we use distilled water, which you can get from a supermarket. And so we're just going to add enough of this distilled water into our solution to form a paste, hence the name saturated paste extract. And one of the things that's important is you can see if I'm mixing up the soil, if there were any controlled release fertilizer prills in here, then they would get broken open and the EC would go up and I would get a false high salt reading. So we need to remove any controlled release fertilizer prills from this substrate before we start mashing it up as we mix it. And we're just going to slowly add enough distilled water in here until we've completely saturated all of the pores. Those are the holes in our substrate. Just going to mash it up in here. And people talk about surface glistening, and if you tap the sample just a little bit on the bench surface, then you're going to start to see a little bit of free water. So that sample is now ready, and I would leave it there. Here we go. I'd leave it there for about 30 to 60 minutes for the solution to equilibrate. And then what I would do is I would take this sample and I would put it through a filter. One of the most practical ways to filter it is with a coffee filter if you're doing it in your nursery. And so you're just going to filter it out and then measure the pH and EC in the filtered solution. Okay? The measurement you would get if you directly measure, put your sensor into this slurry, would be very similar with the filtered solution. It's just that it's tougher on your meters if you're putting it directly into this mix of substrate and water. So that's the saturated paste extract method. You might have seen it's taken me a couple of minutes to get that sample ready. And so if you're doing a lot of samples in your greenhouse and nursery, that really adds up in terms of time. So what some growers will do is they will collect their sample in the same kind of way, right? From the bottom 2/3 of our containers. We're going to combine them into a sample, and they're going to get around a 1/2 cup or so of sample. They're going to compact it down to about the same level as compaction as we had in the container, and we're going to do what's called a 1 part of substrate to 2 parts of water test here. So it's very important, this is very basic, but it's very important, that you measure the water separately from the substrate. Okay? If I just poured water in here and came twice the volume up here, then I'm not going to add as much water as if I measure the water separately, because the substrate takes up space. So 1 cup for substrate and 1 cup for water. I like these cheap plastic beakers. You know, they cost, you can get them from online from a science supply store or similar. You can also just use a cup. Okay? But the nice thing about this is I can read that I know I have 125 milliliters here, so I need to have a little bit more water here. Yep, I've got 125 mils of substrate and 250 mils of water. I'm just going to mix those together, and I'm going to leave that for, again, 30 to 60 minutes, and I can directly measure into that, that sample. Doesn't even usually need to be filtered. There's typically not a big change over that 30 minutes. Okay, so if you're working fast, you're pumping out some quick tests, then you leave it 5 minutes. Frankly, if there's a problem, you're going to see it even after 5 minutes. Okay, so that's a 1 to 2 type of test. 1 part substrate to 2 parts of water. In other countries, they have different amounts of dilution. So for example, in New Zealand, I understand that it's quite often they'll do 1 part of substrate to 1.5 parts of water. In Europe, I've heard of 1 part of substrate to 5 parts of water. And the advantage of the 5 parts of water is just that you've always got a very consistent ratio between the substrate volume, dried substrate volume, and the water, because how much water there is in the substrate is not going to make much difference at the time that you collect your sample. But the reason that the saturated paste extract is used by the lab is that we know pretty consistently how much water we're adding in here. Where the 1 to 2 soil test, there's a bit more variability because it partly depends on how moist the sample was when we took it from our container as to how much water is going to end up in our final sample. A 1 to 2 soil test is much more diluted than a saturated paste extract sample. So the target EC levels with a 1 to 2 are going to be lower because we've diluted our salts than they would be for a saturated paste extract. So quite often we might be looking for an EC of around 1 with a 1 to 2 and test, and we're looking for an EC closer to 2, 2.5 with a saturated paste extract. So these are both soil testing methods where we're taking a sample out of containers and we're mixing them together. We could use that for any container sizes. If we were doing plug cells, we could take the entire cell. However, you can imagine that if you're getting 500 mils of sample to send off to a lab with plugs, you've destroyed a lot of plugs. So there is a non-destructive sampling method called a plug squeeze or plug press, where what we do is within an hour after irrigation, so that the substrate is nice and moist, then we're going to either just push out the soil solution, or if it's a stabilized media such as these paper pots here, we can squeeze out some solution, and we can collect that from at least 10 cells to get a representative sample. And so this is our plug squeeze. We don't do any more dilution than that, and we can measure our pH and EC in that sample. So that would be the plug squeeze. Very good for any propagation trays up to bedding plant flats where we've got small cells. There's another sampling method which is kind of similar to this plug squeeze in some concepts, in that you're displacing the soil solution. Is if we have a container, we can do what's called a pour through. And with a pour through, then what we're going to do is we're going to irrigate our crops within an hour before we take our sample, which can sometimes be a little bit complicated to organize that if you've got different irrigation zones. But it's important that the substrate is moist and it has equilibrated. Because what we're going to do is we're just going to add a little bit of distilled water to the surface of this container. And as we add that water, and we're going to measure out, I'm going to add 50 mils here, I'm checking the volume. We're going to displace a certain amount of sample out the bottom. Get a little bit more. Okay, that's 50 mils. Okay? And so what we're trying to do when we do a pour through is we're just trying to add water at the top, and that head of water, that pressure, is going to push the soil solution from the lower parts of the container out the bottom. Now, if this was too dry, then the solution that we put in the top is just going to channel through the holes in the dry media, and what we're going to sample out the bottom is what we're putting in the top. So that's the reason with a pour through that it's really important that we've got a nice moist media. And one of the things is it's nice and quick. It's not destructive. If we've got a crop like a stock plant, then we don't really have to touch the foliage or touch the soil very much to take that sample. So sanitation-wise, it's good as well. One of the things I find often with pour-throughs is people cut corners. They only take a pour-through out of one plant. You need to combine the solution from several plants together. And again, you're just going to measure pH and EC directly into this solution. It's a little bit more variable than the other testing methods from my experience, but it's very practical and quick to do. Also, if you've got controlled release fertilizer in your mix, you don't have to worry about removing those prills or anything. So so long as you're always taking your sample an hour after irrigation, we get some pretty consistent results. Now, over here I have a stock plant of coleus, and in this case we've got a volcanic rock substrate. So I do a fair bit of work with growers in Africa and Latin America, and this is quite a common type of substrate. There's, in this case, there's no peat or bark or coconut coir in here. It's just pebbles, just small stones. And so one of the things with this type of hydroponic type of substrate, where we're irrigating with a dripper, one of the things that we do is we put some of the drippers into containers to measure the volume and pH and EC of what's getting applied to the crop. And then we have a funnel or some other catchment arrangement to get the drainage coming out of the bottom. We typically look for a target leaching fraction, which would be the drainage volume. Let's say 200 mils draining out of 500 mils being applied would be 40% leaching fraction, for example. So we know how efficient our watering is and how much through flow we have. But that quart drainage volume, we can use that to test pH and EC, and it's very similar in terms of our interpretation with how we would be interpreting a pour-through. So that drain type of an approach is very similar to a pour-through test. So we have multiple options here on how to take a soil test. There are certain advantages and disadvantages with different types of crop. The important thing is that you have somebody who's very detail-oriented. They have a good quality meter. They calibrate the meter. They follow the protocol in a consistent way. Don't try to make up your own kind of soil test. Use a testing method that's standardized and where you've got other growers, other consultants, extension agents that you can refer to get some help with interpreting the results. Thank you very much.